So in this lecture, we will talk about another class of molecules known as lipids. So lipids are an umbrella term, and there's a wide array of different types of molecules that can go in under the category of lipids. But most or of all of these molecules are unified by the fact that a lot of lipids are soluble in organic solvents, like your chloroform, methanol, or hexanol. Very rarely do we find water-soluble lipids. So the most simplest lipids are fatty acids. Fatty acids are just long-chain carboxylic acids. They have an acyl chain, which is hydrophobic, and a head, the carboxylate, shown here, which is charged at physiologic pH, and that's negatively charged. So the whole molecule, the whole fatty acid itself, is what we will call amphiphilic, or some textbooks also call it amphipathic. But amphiphilic basically means the single molecule has a hydrophobic part to it, and another part is hydrophilic. Now, another thing I want you to realize about lipids, besides the fact that they are soluble in organic solvents, one thing is they take a wide array of different structures. So a steroid, which in and of itself is an umbrella category, looks very different than a fatty acid, which looks very different than a triacylglycerol, which is a fat. And also lipids make up a major constituent of our membranes. Those are sphingolipids and also phospholipids. Steroids, as we mentioned before, are mostly signaling molecules, such as cholesterol, but they also play a role, specifically cholesterol, in regulating the fluidity of the phospholipid bilayer, which in turn is composed of phospholipids. Vitamins, a whole new class, another umbrella term within the larger realm of lipids. So these will assist in different functions within the cell, bone absorption or calcium absorption. Vitamin K is important in blood coagulation. So big, huge umbrella term here, lipids. And individual lipids may have very, very different structures. An important thing you should know about lipids, particularly with the phospholipids and the phospholipid bilayer, is this concept known as phase transition temperature. So lipids can exist in a gel form, which is kind of like a gel matrix, or they can exist in a solution form, which is like a liquid, where all the molecules are diffusing and motion is like a liquid, or they could be all clumped together like a gel. So this phase transition temperature is exceedingly crucial in our cell membranes. Keeping it in fluid, you may have heard of the fluid mosaic model. That's where phase transition temperature comes in. So here's a review of fatty acids, particularly their nomenclature. So long chain carboxylic acids, we can actually divide this molecule into two parts. One part we're going to call the acyl chain, and then that little polar head group here usually is polar and has a negative charge affiliated with it. So these are amphiphilic molecules, sometimes also called amphipathic molecule, in the sense that you can have a polar end and then a hydrophobic long tail, kind of like a lollipop, except you have two sticks to that lollipop. Some fatty acids have a double bond. So the double bond is cis. So that's an important thing to understand, is that the fatty acids, their acyl chain more specifically, will be in the cis configuration. And the final notion is nomenclature. So what we have here are 18 carbons, if you count the number of carbons. The designation of the double bonds, if you count from the carboxylic end, so 1, 2, 3, 4, 5, 6, 7, 8, 9, that will be a delta 9. So oleic acid will be C18 delta 9, 18 being 18 carbons. You can count it from the terminal methyl end, so 1, 2, 3, 4, 5, 6, 7, 8, 9. Counting from the terminal methyl end, that gets the designation omega. So oleic acid is C18 omega 9. You may have heard of omega fatty acids and their importance in nutrition and their abundance in some foods. That's where omega fatty acids come from. Another thing is saturated versus unsaturated fatty acids. So saturated fatty acids, as shown here, stearic acid, is saturated with hydrogen, so no double bonds. The minute you add a monounsaturation point or a diunsaturation point or polyunsaturation points, that is when you get unsaturated fatty acids. It could be monounsaturated, diunsaturated, or more than 2 polyunsaturated. Now, phase transition temperatures are going to be important here. So phase transition temperature for a saturated fatty acid, we would expect to be high. So the temperature at which the molecule, the fatty acid, will either be in a gel form or in a solution form. Now, it's worth noting the acyl chain in the liquid form is all loose, loosey-goosey. We have a lot of rotation across the axis, the long rotational axis of the long acyl chain. In a gel situation, the acyl chains are packing, and they're kind of packing like sardines. Okay, that would be in the gel state. It reflects itself in the way the molecule is, kind of like a gel. That's below the phase transition temperature. The minute you add double bonds, you dramatically reduce the phase transition temperature. Why is that? It has to do with packing. So that double bond presents sort of a kink into the packing arrangements. So the acyl chains do not pack so succinctly, so nicely. They're sort of kinks in the packing arrangements. So that's going to disrupt the packing, but it will also lower the phase transition temperature in the sense that it takes more temperature, it takes more heat to disrupt acyl chains that are packed very close together. The minute you add a double bond or multiple double bonds, it takes less heat for them to disrupt that pack because they're not packed tightly. The double bonds add little kinks into the packing arrangements. So add a double bond to this long acyl chain, you lower the phase transition temperature. And that's really shown here. Let's take a look at, for example, palmitic acid. So here's a free fatty acid that's very important, by the way, in your lung. So 16 carbon, 0 double bonds, saturated fatty acid, and the phase transition temperature is 63.1 degrees C. So above 63.1 degrees C, we're going to get a solution. Below 63.1 degrees C, palmitic acid will pack like a gel. Above that, it's kind of going to have free molecular motion, okay, at that high temperature above and beyond. Now, right at 63.1 degrees C, right at the phase transition temperature, you're kind of 50-50 gel and 50 solution. So at body temperature, palmitic acid will be a gel at room temperature. So in the case of the lung where palmitic acid exists, not a problem. Now, the minute you add a double bond, so I'm not liking this N-7 here. You can look at the footnotes. But the minute you add a one double bond, or monounsaturation point, delta 9, okay, delta 9, the phase transition temperature moves from 63.1 degrees and goes down all the way to minus 0.5 degrees C. Okay, so that is definitely at room temperature a solution. So palmitoleic acid will be a solution at room temperature, okay, because room temperature being about 25 degrees or so. So the minute you add a double bond, you're really dramatically reducing the temperature at which this will transition based on packing of the acyl chain. You dramatically reduce the temperature at which it goes from a gel transition to a solution transition. And then you can see here, the more double bonds you add, okay, the lower and lower and lower the phase transition temperature. Okay, so here DHA has 6 double bonds. Minus 44 degrees C is the phase transition temperature. So you got to get it below minus 44 for this DHA polyunsaturated fatty acid to gel. those six double bonds really mess up packing. Very distorted, and it doesn't take a lot for it to get into a liquid state. All right, so we will build upon the simple fatty acids, and what we will do is we will take the fatty acids and join it together with glycerol. And we're beginning to take the shape of a phospholipid. We really need to add a phosphate in that head group. And then we have the major constituents of the membrane. So usually in eukaryotic membranes, the fatty acids are esterified. Okay, esterified meaning there's an ester bond. So the esterification occurs if you have 3 fatty acids esterified through the glycerol backbone. We call that a fat, triacylglycerol. So this is not in our membranes. These are bad molecules in the sense that they tend to store in adipocytes, but very great source of energy. You can get up to 100 to 120 ATPs per hydrolysis and metabolism of a free fatty acid. So a lot of energy is involved in metabolizing a fatty acid. Problem is cardiac cells and muscle cells will do that. The brain doesn't really undergo beta oxidation, which is the metabolism of fatty acids. Usually the brain relies on glucose or other sugars as its source of ATP. Fatty acids have a difficult time crossing the blood-brain barrier. So typical fatty acid, 3 of them esterified to a glycerol backbone, and here we have a typical fat. Right? So here this acyl chain has a double bond. This fatty acid is diunsaturated. Here we have a saturated fatty acid. Again, ester bond to glycerol, triacylglycerol, a fat. Okay, the nomenclature for this particular fat is shown here. And here you see the ester bond. And here you see the 3 acyl chains. All right, now let's talk about membranes. So any basic biology course talks about the membranes of our cells and how they are a phospholipid bilayer. So these are the constituents of that bilayer. It's a glycerol phospholipid. So as before, we're going to have the ester bond with the glycerol backbone. But on this 3rd carbon, we're going to add a phosphate and a head group. Now, the minute you do that, this glycerophospholipid takes the shape of a big head, which is charged, usually negative charge, but it can be positive, and then 2 tails, okay, 2 tails that sort of lounge around. Okay? And that is how our membranes are associated in a bilayer setting. These acyl chains pack. And the packing is autonomous, meaning it's self-assembly. It happens on its own. The head groups interact on its own. Your phospholipid bilayer, your membranes, really is just all self-assembly. Right, so here's our glycerol 3-phosphate. We're going to esterify with fatty acid 1 and esterify it with another fatty acid. It could be the same, it could be different. Fatty acid 2, it could be saturated, it could be unsaturated, and all of this comes from glycerol 3-phosphate. So by the way, glycerol 3-phosphate actually is produced in our body as a part of our metabolism. So people have a lot of fructose. Fructose gets taken in, particularly the liver, and one of the metabolites that fructose can get broken down to is glycerol. Right. So here's our backbone of a phospholipid. Just want to give you some information here. If you take a signal transduction course or cell biology course, you find they often talk about lipases. So these lipases will actually cleave at the ester bond, and that's a way to liberate free fatty acids. One phospholipase is pretty interesting. Phospholipase C will cleave at this specific bond, and it will release second messenger molecules. So you may have heard of cyclic AMP or diacylglycerol. And we will not study them in this class, but those are second messenger molecules that aid in signal transduction. So these phospholipases have a very important role in the vicinity of the cell in terms of signal. There you see our glycerol backbone. This is going to be a common theme here. The esterification of 2 of the carbons of glycerol, and that 3rd carbon of glycerol, what was formerly glycerol, will have the phosphate head group. So here's how we tend to visualize a phospholipid. Now we're talking about glycerophospholipids. A little head group here, and then 2 acyl chains. And what you see here is this protein trying to attack one of the ester bonds. A lot of diversity and variations in this head group region. So really this diagram or this table from your textbook is showing the vast array of different head groups. The simplest X here would be hydrogen, and that would be phosphatidic acid. So all of these are constituents of your membrane. Some are a little bit simple, like phosphatidylglycerol. Some are more complicated. Here you see a sugar, phosphatidylinositol. This is part of your membrane. A lot of these will have different charge impacts. So if it's positively charged, usually they're on the surface, outside of the cell, or on the inside of the cell. The hydrophobic interior are your acyl chains. So a lot of different diversities. In fact, this whole lecture is basically going through some of the different types of lipids that we see. But we'll also talk about vitamins and other assortments of lipids. There's our glycerol head group again. Okay, this once was formerly glycerol, acyl chain from fatty acid 1, acyl chain from fatty acid 2. Phosphate gives the head group a negative charge component, and the X can take on a whole suite of chemicals. So here's our first type of glycerophospholipid. 1 steroid, 2 oleol, 3 phosphatidylcholine. So the head group here shown in green is a choline head group. Here is our phosphate. Here's our glycerol backbone, and here are 2 fatty acids. Notice the double bond here. Okay, provides a kink. Okay, so here's our head group. Here's our 2 little acyl chains. The double bond provides a kink. So sort of the dissection of the individual parts of the glycerophospholipid here. Okay. Carbon number. This is oftentimes we'll call this an SN carbon. So this would be SN1, SN2, and then SN3. So more membrane biologists will say that stearic acid is esterified to the SN1 carbon, oleoic acid is esterified to the SN2 carbon of glycerol, and then the head group is attached to SN3. So how about we look at another type of phospholipid? These are plasmalogens. So the plasmalogens, well, they do have that glycerol backbone. Plasmalogens do have that glycerol backbone, which you see here. But here we don't have an ester bond, but we have an ether bond. And then here we have our normal ester bond. So we have an ether and then an ester, and we have the phosphate head group as before. So a little bit of a variation of our glycerophospholipid are these plasmalogens. Okay. Ether bonds actually, I don't know if you care to know this, but ether bonds are actually much more stronger, harder to break than an ester bond. So here is another type of, well, we'll call it a phospholipid, but it's called sphingomyelin. This is very much enriched in nervous tissue. And again, look at the general, okay, I'm speaking very broadly here, general organization. A head group here, and then 2 acyl chains. Okay, here the acyl chains are kind of flat and floppy. Again, no bends and kinks. So it kind of just hangs loose here. No bends and kinks because all of these fatty acids are saturated. But we have a nitrogen here to which we have attached our fatty acid. Okay, this would be palmitic acid. By the way, this is called an isopeptide bond, C-double-bond-O-N-H. It is a peptide bond, but it's not joining together amino acids. So outside of amino acids, we call it an isopeptide bond. But we don't have an ester or ether bond. So this fatty acid is not esterified. So sphingomyelins are distinguished from glycerophospholipids in the fact that we have a non-glycerol backbone. We have what is called a ceramide backbone. And so a non-glycerol phospholipid here. Here's our ceramide entity, again, non-glycerol backbone. Well, glycerol backbone or non-glycerol backbone take on the same conformation, head group and 2 acyl chains. Head group, 2 acyl chains. So sphingomyelin is very much found in the myelin sheath of nerve fibers. All right, gangliosides, all right, non-glycerol backbone, ceramide backbone, okay, and attachment to sugars. All right? So a whole bevy of sugars here. These sugars actually are situated outside of the cell and very much serve as receptors for good guys and bad guys. Okay, so the bad guys would be bacterial toxins. The good guys would be maybe other cells. Definitely a lot in the literature on gangliosides, a lot implicated for these in spreading cancers. If you've done the lab on biomedical bioinformatics, you can do a PubMed search on gangliosides and you'll see their relationship. Their relationship to health and disease. But we have a ceramide backbone. Here are our two fatty acids inside the ceramide backbone. We do have a bunch of sugars that are attached, which are sort of binding sites for other molecular entities. Also binding sites for pathogens as well. So gangliosides have a good and bad component situated with them. It's a lipid. We know a ganglioside has that ceramide backbone, just like sphingomyelin. A ganglioside has these sugars attached to it, but then again, look at the same conformation here in these ball and stick models. A head group, a head group, a head group, and then acyl chain. Okay, two acyl chains. Okay, here's an acyl chain with a double bond. Here's an acyl chain. All right, let's talk about cholesterol. So cholesterol is part of your membrane, and it is a lipid. Well, it is a lipid that we will call a steroid. So another big class of molecules. A bigger umbrella is lipids, sort of the smaller umbrella under lipids is steroids. So here we're talking about cholesterol. In the milieu of our membrane, cholesterol is an amphiphilic molecule. Okay, this OH can attach itself to fatty acids. This OH provides some necessary polarity to the membrane, giving it some amphiphilic character, part hydrophilic and hydrophobic. And this four-member ring, fused four-member ring, is hydrophobic. So we would expect that to sit at the hydrophobic interface, interacting with the acyl chain. So regulation of membrane fluidity is an important role in cholesterol. They are part of our membranes, and they're very important. And so maintaining the membrane so it doesn't gel up. The worst thing that could happen to your membranes, or anybody's membranes for that matter, is that it gels. And if it gels up like jam, it has that type of viscosity, and if it has that type of material, a lot of things can go wrong in the cell. So cholesterol sort of serves this function of keeping everything fluid so that proteins and other entities can sort of swim in this phospholipid bilayer. Cholesterol, not just in the membrane, but it has a role in metabolism. So it's a metabolic pathway that produces other types of steroids, such as the androgens and estrogens, produces anti-inflammatory agents. So very, very important molecule. Definitely gets a bad rap because of its association with plaques and arteriosclerosis. We do have a good cholesterol and the bad cholesterol. Bad cholesterol is what you get from food. Good cholesterol interacts with our plasma membranes, making it more fluid. Also, the good form of cholesterol is important in these metabolic pathways. So 30 to 40% of your plasma membranes are cholesterol. So with your glycerophospholipids, with your gangliosides, if you're a nervous cell, nervous tissue cell, with your sphingomyelins, we're having something that's very fluid. Okay, and they're all self-assembling. All right, let's go through vitamins now. This is the second to last part of the lipids. So big umbrella, vitamins, underneath the even larger umbrella of lipids. So vitamins have a diverse array of function. I want you to look at the structure of these. These structure, particularly of vitamin D, very, very different than fatty acids, very different than your gangliosides, very different than your sphingomyelins, is it not? Here, this vitamin D3, looks more like a steroid. And so here's vitamin D. We should learn that it's just a lipid. Functions in the cell, probably bone resorption, probably calcium absorption, mineralization. There's a vast array of functions for vitamin D and the different families. Coenzyme Q is a lipid. and it is not a vitamin per se, but what coenzyme Q is, and you can actually buy this in some supermarkets, nutrition stores. Coenzyme Q is very important in the electron transport chain. So coenzyme Q literally carries electrons from complex 1 to 3 and 4, or from complex 2 to 3 and 4. This happens in the inner mitochondrial membrane. That's in communication with the mitochondrial matrix. Coenzyme Q is that facilitator of electrons. It undergoes what's known as the Q cycle, so that the electrons are sufficiently funneled from one complex to the other in the electron transport chain. Coenzyme Q is a lipid. It's soluble within the membrane interface, so it's very important that it's fluid, our plasma membrane, particularly in the inner mitochondrial membrane where electron transport happens. It's important that it's a fluid because soluble in that fluid phase is coenzyme Q, an electron carrier. And look at its structure, it's a lipid. Vitamin A is found in the rod and cone cells in the back of the eye. It's a lipid. Here's the structure. Again, points of these slides are not for me to, pick on individual functions, but telling you that, hey, look, these are lipids, okay, vitamins and fatty acids and cholesterol, but they don't look alike. Okay, structurally speaking, they don't look alike. They are soluble in organic solvents, and they have very, very different functions. Obviously, this is important in visual transduction, whereas maybe a cholesterol would be important in membrane fluidity. Okay, but these are all lipids. And here's an interesting lipid, vitamin K, under the category or subcategory of vitamins. Vitamin K is something that we do not produce. Vitamin K is something that we get through our gut microbiota or the gut flora. So vitamin K is involved in blood coagulation, complement cascade, allowing blood to coagulate so that we don't bleed. Hemophiliacs are deficient in this cascade, known as the complement cascade. So when they bleed, it becomes a big problem because the blood doesn't stop. Vitamin K's role in that small role in this huge process is to carboxylate glutamate. So it's performing a catalysis. Look at the structure. Okay, very different from these other guys that we've been studying in this PowerPoint. And it's a vitamin. Vitamin E, again, a lipid. Look at the structure. Very different. Probably the only thing we can say in relation to the other lipids is that it is soluble in organic solvents, okay, which is one hallmark of lipids. So vitamin E associated with the membrane and has its different functionalities. All right, our final lipid here is another category. So we talked about vitamins, we talked about glycerophospholipids, we talked about cholesterol, steroids, we talked about gangliosides. Now let's talk about another category, the eicosanoids. This is metabolism of the C20 fatty acid, arachidonic acid. These act sort of locally, kind of like a hormone, cells that secrete it, and they symbolize pain, or they allow for the perception of pain in different tissues. So your body metabolizes arachidonic acid into these three parts: prostacyclins, prostaglandins, and thromboxanes. Thromboxanes, prostaglandins, and prostacyclins are involved in generating pain. They act very locally. Aspirin inhibits the metabolism at this site, PGH2 synthase. Aspirin also inhibits at another enzymatic site known as the COX enzyme, COX1 and COX2. Aspirin is a competitive inhibitor of that enzyme that's involved in pain perception. So here's a fatty acid that produces a whole other class of molecules that are involved in pain.